Great. So we might have a few latecomers, but let's begin. Welcome. My name's Nathan. Welcome to the first seminar in the series for 23-24. We've got a lot of great speakers lined up. So setting the bar today is going to be Somak Dutta from Iowa State University. I'm very pleased that you can speak to us, Somak. And he's going to be talking about Bayesian screening and variable selection in ultra high dimensional linear regression. So over to you, Somak.
All right, thank you, Nathan. Thank you for your kind introduction, and thank you for the invitation. It's a privilege to be the first speaker of this seminar series. And I was told by Nicholas that maybe the title and abstract in the announcement was a bit different, so your prior expectation and the posterior experience might be a bit different. So I'm here at the Iowa State University, and I hold a unique position that I do a lot of collaboration. My position is entirely funded by the College of Agriculture and Life Sciences. So I do a lot of collaborations with them, and many of my statistical research actually arise from problems that come from agriculture. So this is a set of works on ultra-high dimensional variable selection, screening and selection came from several requests from the agronomy department. And these are the theory and method part of these works are with my colleague Vivek Roy, whose name somehow is not here appearing. So it's Vivekananda Roy, the one person on the right, and two postgraduate students, PhD students, Dongjin Lee and Rune Wong. Dongjin's at Wells Fargo after she graduated, and Rune joined Google. So kind of, we have to train a new set of PhD students going forward. All right, and all of my applications collaborators are from either the agronomy department at Iowa State, or the animal science, or UNL and Montana State. So I won't be able to talk about all the applications, but I will just briefly talk about one application that kind of shows the, illustrates the methodology. So I will talk about this particular genome-wide association study problem, where we have close to 4,000 inbred lines from 282 founding lines. So if you don't know about these inbred lines, so these are 282 corn or maize varieties founding lines. They're a pretty diverse set of genotypes. And they are randomly crossed between themselves, and these offsprings are inbred for a few generations so that their alleles become homologous in most of the locations after a few generations. So this 4,000 inbred lines could be thought of as a random sample of the children that might come out of these 282 founding lines. And what we have is a vector of response values. For example, the kernel protein, the protein content in the corn or maize kernels, or starch, or fat contents. And we have this giant matrix Z of marker information. So this is an N by P matrix, where P is the order of 500,000. And every entry, each locus, like every column of the matrix Z, are encoded into 0, 1, but they actually correspond to allele values. So they can have only two possible allele values, either like A and C or A and T. So one of them would be called the major allele, which is more prevalent in these inbred lines, and the other one, which is less prevalent in the inbred lines for a particular marker, would be called a minor allele. So generally, the more prevalent allele, the major allele, is coded as 0, and the less prevalent allele is coded as 1 to store the matrix Z in a sparse format. And not all the locus are equally prevalent. So generally, loci with their minor allele frequency less than 5% are considered to be not very informative. So they are typically dropped. And that results in close to 500,000, more than 500,000 marker locations. But the scientists believe that among all these marker locations, only a few should be important. That's their assumption, and in the statistics literature, it's called the sparsity assumption. So our goal is to find these important markers that are associated with this response value, with this phenotypic response. So that's basically finding a few needles in the big haystack. There are other interesting and important plant phenotypes that we have also been working on. There are James Snavell's lab has a lot of close to 100 or 200, 110 maize phenotypes that are related to agronomic traits or cellular or biochemical disease or flowering time and all those different traits. We have measurements on vitamin B contents because that's a very important trait. We would like to increase the vitamin B content in maize. Or the bottom right panel, which we've been working on, I think to me it's very interesting because I saw the effect. So in 2020, right after, well, not right after, but after the pandemic began, during the summer, a big storm hit Iowa. It's called derecho. It decimated the crops, all the crops, and created a substantial root lodging. So most of these corn plants, many of these corn plants fell on the ground. So the scientists went back to the field and then they measured kind of how these plants tilted, and they coded those root lodging using some scores. So the scores are basically interval censored. So if a plant is standing tall, like standing around and standing straight, it's coded as root lodging score 1. If the angles between 60 and 90 degrees is coded at 2, and so on. So higher root lodging score means the plant is the particular genotype is more susceptible to root lodging. And so their goal was to understand how we can breed future corn varieties that are resistant to root lodging or to abiotic stress, so high wind damage. So what is done is, field trial experiments. Typically, this data come from field trial experiments, and we don't have a very many genotypes. So as you saw, there were like 4,000 genotypic lines, and so that's kind of what we have to work with. But and we measure all these different traits, typically through spatial or through design of experiments, field design of experiments, and we do adjust for the design and for example, spatial variability to finally compute the response values that would go into our association mapping. So we compute the genotypic blue. So we have Y as an n by 1 vector of continuous response values, for example, which are the genotypic best linear unbiased estimators. And we have this matrix Z, that's an n by p matrix of covariance, and in our setup, P is very large. So existing methods for identifying the important SNP markers are, in general, the variable selection are penalized. There are two types of methods generally, broadly speaking. The first set of methods penalize either the effect size or the model size. So because we have more variables than observations, some penalizations are needed, but either the effect size of each variable or covariate is penalized or the model size is penalized, but seldom both are penalized simultaneously in the first set of methods. So for example, lasso, SCAD penalty, elastic net, this penalizes the effect sizes, whereas L0 penalty, that penalizes the model size. On the other hand, there is this approach that integrates the penalties on the effect size and the model size in a single model. And this dates back to Box and Meyer in 1986, who considered orthogonal design. So in their applications, the number of the covariates were not very high dimensional, and the columns of the matrix Z were orthogonal to each other. Later, George and McCulloch kind of pioneered the set of research, like the variable selection set of type of research, and more recently, these research have been extended to the ultra high dimensional setup. So Narisati and He published this paper in Annals of Stat in 2014 that talked about kind of the posterior consistency results in ultra high dimensional setup. Then Drokova and George extended the EM approach to Bayesian variable selection in the ultra high dimensional approach. And there are other methods by Shin et al. On using non-local priors on the effect sizes. So all these methods, they claim to work in ultra high dimensional settings where the number of variables is exponential of little o of n. So in other words, logarithm of p is growing slowly with n. But most of them suggest preliminary screening, that is, reduce the number of covariates, reduce the number of columns of Z to a manageable one for mostly computational purposes. This is called screening. So the frequency solution to screening reduce the number of predictors from e to the power little o of n to big O of n. Finally, in JRSSB published a paper called the shear independent screening that keeps variables with largest absolute marginal correlations with the response. This is probably one of the most cited screening methods. Then these ideas were extended to keeping variables with largest marginal distance correlations or keeping variables with largest marginal rank correlations. And more recently, Wang and Leng published this paper that keeps variable based on minimum norm solution. So we minimize the least square norm subject to least square error subject to the norm constant, so that there are many solutions. The one with the smallest norm creates an ordering of the variables that can be proved to be screening consistent, that keeps the important variables at the top with probability tending to 1. And this solution avoids avoiding looking only at marginal relationships between the response and the covariates. All right, so let's look at the SIS, the shear independent screening that screens using the largest absolute marginal correlations. So it's been cited more than, I think, 2,800 times now, and it's routinely used by scientists. We compute the Pearson correlations between the ith covariate and the response, and then keep, for example, the top dn variables with largest absolute values of these marginal correlations. So finally, it suggests n over log n as the screening number. However, this method has strong assumptions about the dependence structure among the covariates. So in general, if there is correlation among the covariates, then this method may not work very well. So for example, in this small simulation study, the rows of the matrix Z, actually not X, is generated from a multivariate normal distributions with a compound symmetry correlation structure, covariance structure. And even if the number of variables P is not very high, let's say it's only 2 or twice the number of observations, then beta can be chosen, or beta can be chosen so that at the population level, what happens is the correlations between the response and many of the important variables is actually smaller than the correlation between the response and many unimportant variables. So in fact, in this particular case, the correlation between the response and all the unimportant variables is 4 times the correlation between the response and some of the important variables. And because of this, as n goes to infinity, it can be shown that these actually important variables surely fail to survive screening. That, for example, when the sample size is around 10,000, by this time, the variables x1, x2, x3, x4, which were actually important, fail to survive screening, that they are ranked very low more than 96% of the times. When p scales at the order of n squared, this problem is more severe. I mean, the failure rate converges to 1 even faster. So we were thinking of, because we are interested finally in a Bayesian method. So we were thinking, like, how can we construct a Bayesian method for screening that is based on the same model? So it's in some sense, it's faithful. The screening method is faithful to the Bayesian variable selection method. So we will propose our the hierarchical Bayesian model and the Bayesian iterative screening now, and then talk about the screening embedded variable selection, followed by data analysis. All right, so let's say we have this in the Bayesian setup. What we do is we assume a latent indicator variable, gamma. So gamma is a p/1 vector of latent binary variables indicating the inclusion or exclusion of the variables. Okay, so then what we assume is that, let's say x is the scaled version of z, so it has mean 0 and common standard deviation, unit standard deviation actually. And we assume that the response y, given all the coefficients, intercept, the error variance sigma squared, and this latent binary vector gamma, depends linearly only on the subset of the matrix X for which gamma is 1. Okay. So X gamma is the subset of matrix consisting all those columns of X for which the latent binary variable takes a value 1. And then we have this hierarchical structure where we assume beta j has independent normal structure with mean 0 and the variance that is gamma j over lambda and sigma square. So this is a bit important. So if gamma j is 0, this is normal 0, 0. So we assume a priori that for an unimportant variables, beta j is exactly equals to 0. On the other hand, when gamma j is 1, that is, if the variable is important, then beta j has a normal distribution with mean 0 and variance sigma square over lambda. Generally, lambda is taken to be very small, and we will see some theoretical results, so that it's a kind of a flat prior. The variance would be large, and the prior is more or less flat on the real line. And we have a flat prior, a non-informative prior on the intercept and the marginal on the error variance. And we assume that these latent indicator vectors, gamma j, given some prior inclusion probability W, are IID Bernoulli with a success probability of W. Typically, W is small, so we assume a priori that only a few markers are important. And this lambda and W are assumed to be known non-random functions of n and p. And so our goal is to find the posterior distribution of gamma. Now, that's a very high dimensional distribution. It can take 2 to the power p many possible values. But we would like to find out kind of, the important variables, so gamma I with high posterior probabilities in some sense. All right. So actually, the posterior distribution of gamma is available up to a normalizing constant in an analytic form. So the log unnormalized posterior distribution, log posterior distribution actually, sorry, is it's a constant minus the half of the log determinant of a matrix X transpose gamma, X gamma plus lambda I, minus half n minus 1 log R lambda, where R lambda is kind of the ridge version, ridge regularized residual sum of squares. And then there's this penalty part, the part arising out of the model size penalty. So here in this approach, the effect sizes are penalized through the normal prior with the penalty parameter lambda in some sense. And the model sizes are penalized through this prior inclusion probability, so this Bernoulli prior. All right, so screening method. So let's suppose we do not a priori we assume anything to be important. So we start with all the variables as unimportant. And then we compute the unnormalized probability, the probability that the ith marker is important given that the rest are unimportant. And this can be shown to be available in analytically, up to a common constant. This can be computed analytically. And so we say that, let's say V1 is the variable that maximizes this probability. Then we say V1 is the first proposed variable. And we take a decision. So if including this variable, if the posterior probability of this singleton set V1 is more than the probability of the model that has no variable that's important. If it is bigger, we continue. If it's smaller, then we say, maybe none of them is important. So let's just stop. But once, let's say, if we do include V1 in our method, in our bag, then what we do is we compute the posterior probability that all the other, like each of the other, for each of the other markers, we compute the probability that it is important, given that V1 has already been included, and let's say the rest are not important. And then we maximize that probability. Let's say V2 is the marker that has the highest posterior probability of being important, given the V1 was already important and the rest are not. Then V2 is the second proposed variable after accounting for the effect of V1. And if including V2 increases the posterior probability, we continue, but we stop if including V2 actually reduces the posterior probability. And in general, after we have included the first, let's say, k variables, we check for importance of each of the other remaining variables one at a time, after accounting for the facts that several variables have already been included in the screening model. And these are all computable up to a normalizing constant, and we maximize it. And then we proceed. So if including the k plus first variable increases our posterior probability, we continue, but stop if it reduces the posterior probability compared to the already included ones. So this is a sequential decision. It's a greedy decision process. And it has screening consistency properties. That is, let's say our true model is actually T, that, let's say T is a little of n. So the number of markers that are important is scales at a lower order of the lower order with the sample size. And let's say we have, even if we constructed the method assuming the responses are Gaussian, but let's suppose for now that the errors are misspecified. That is, the errors come from the normalized error comes from distribution f with q exponential tail. So what is this q exponential tail condition? So we say that a zero mean distribution f is said to have a q exponential tail if there exists a function q from the positive half or non-negative half of the real line to r, such that for any n bigger than or equal to 1 and eta 1 to eta n that are IIDF, and L that has unit length, and zeta bigger than 0, the probability that the linear combination of all these random variables coming from this distribution f, the probability that the absolute value is bigger than zeta, decreases exponentially, decreases e to the power 1 minus, it's smaller than e to the power 1 minus q of zeta. So when f is normal 0, 1, q zeta is our usual zeta squared over 2. When f is sub-Gaussian, then q zeta is a constant times zeta squared for some constant cf bigger than 0. When f is sub-exponential, q zeta is the tail decreases slowly, so it's a constant times zeta. And if it's even slower, so if only first 2k moments of f are finite, then q zeta is something like twice k times logarithm of zeta. And under this assumption and some more on the regularity, like effect size and so on, some mild conditions that are also included in the whole paper, the frequency screening papers. We can prove that this process has a sure screening property, in the sense that if tau is the random stopping time when the posterior probability decreases, it's a sequentially increasing probability, but first time it decreases, we stop. If that's the random time, stopping time, then under some mild conditions on the design matrix and tuning parameters lambda and w, the probability that the first script t many variables include the important variables converges to 1 as the sample size goes to infinity. And this is in the kind of the ultra high dimensional setup. So that's kind of the screening property, sequentially checking variables one by one. But what we want to ideally, at the end, what we want to do is that we would like to select only the truly important ones. So the screening reduces the number of predictors from e to the power little o of n to big O of n. But the final goal is to identify the truly important variables t. And what we want is also that the Bayesian model to have stronger property, that the posterior should concentrate on the true model instead of just containing it, which might create some false positive discoveries. So we want the posterior to concentrate on the true model. And the natural question is, can we incorporate the screening in the variable selection instead of a two-stage approach? So instead of first reducing the number of covariates and then fitting the Bayesian model on top of it, can we just do two things together? Right. So that's what we kind of worked on. So we have this model selection consistency first, that if let's assume that the logarithm of the number of variables scales slowly with the sample size, and we have some red conditions on the tuning parameters lambda. Let's say lambda is lambda goes to 0. We want the lambda to be small because it's a prior precision. So lambda goes to 0 means the prior distributions, prior variance go to infinity. So it's kind of gives a flat prior on the effects on the coefficients. And we assume that it converges to 0 at the rate of sample size divided by p square plus a little bit for some delta bigger than 0. And we assume that the model is kind of misspecified from our prior expectation. So we assume that there is this true model t. So the response is the intercept plus the effects of the true variables. But there is also a small effect of the unimportant variables. Okay, there is this model T complement. The model T complement has an effect that the total norm of the L2 norm of xt complement times beta T complement, that can go to infinity, but that can go to infinity at a big O of square root of log p. So the model is somewhat misspecified because in the prior, we assume that the coefficients of the unimportant variable is exactly 0. And the true model, it needs to have a bit of effect, right? So that we are able to capture. So essentially what this tells is that P gamma is the projection matrix corresponding to a model gamma. So I minus P gamma times xt beta T squared, that you can think of as the the regression, the population version of the regression sum of squares. And we assume that the regression sum of squares goes to infinity faster than a logarithm of the number of variables. For all model, sorry, I'm wrong. I minus p gamma times xt beta t, that's the population version of the residual sum of square, excuse me. That's the residual sum of square. And we assume that if we miss a truly important variable, then the residual sum of squares go to infinity faster than log p. Okay. So if you give me a model gamma, which is not too large, and it does not contain the true model, so that it misses an important variable, this is kind of the residual sum of squares, population version of the residual sum of squares, and it goes to infinity faster than the logarithm of the number of variables. And that's kind of the thing. And we have, smallest non-zero eigenvalue of x gamma transpose x gamma of all these kind of models. Well, that can go to zero. It's unlike the assumption of the zero independent screening, where this there's a bound on these smaller eigenvalues, but we relax it. We assume that it can go to zero at a rate of p to the power minus kappa, where p is the number of variables and kappa is some positive constant. And we assume that the true model is kind of identifiable in the sense that the eigenvalues of xt transpose xt over n are bounded both below and above. Under these assumptions, if sigma square, the observation error variance, is known, then we can prove that the posterior probability of the true model given the data and sigma square, it converges to 1 as the sample size tends to infinity. So the posterior probability concentrates more and more only on the true model, true set of variables. So a few remarks. This results, in some sense, generalize the results in Narisati and her. They have a very similar set of assumptions, but they assume that the effect of the unimportant variables is kind of bounded. So it's very, very small. So the norm, L2 norm of xt complement times the coefficient, so beta t complement, it's bounded even if n goes to infinity. So we do relax that assumption. So we say that the effects of the unimportant variable can diverge, but not too fast. And it turned out actually that Yang, Wainwright, and Jordan, they also obtained similar relaxations on the total effect of the unimportant variables under a different prior, G priors on beta. But they had made this a bit strong assumption, I would say, that on the minimum eigenvalues of x gamma transpose x gamma by n, they assumed the minimum eigenvalues of this type of matrices where gamma is not too large models. The minimum eigenvalues are bounded away from 0 for all gamma, where mod gamma is less than order of n over log p. This is a bit, can be criticized on the ground that, in our setup, n over log p goes to infinity. So remember, log p scales in the ultra-high dimensional setup, log p scales as little of n. So n over log p goes to infinity. Now I can understand that, the smallest eigenvalues of boundedly many things can be bounded away from 0. But when the number of things or the number of random variables go to infinity, then it can be proved that the minimum converges to 0. So, you can just take one column at a time. Let's say let's look at small models of size 1. And so, xi transpose xi by n, the infimum of those converges to 0. So this can be criticized this assumption can be criticized on that ground. So we have relaxed. We don't make any assumption that, well, we assume that these eigenvalues can go to 0 at a rate p to the power minus kappa for some kappa positive. All right. So when sigma square is unknown, which is generally most of the practical cases, so sigma square is unknown. We do need a bit of further assumption that assigns a prior probability of 0 on a set consisting of unrealistically large models. So if sigma square is unknown, we have to assume a priori that models that have actually size more than n over log p have a prior probability of 0. So that's not too inconsistent with our assumption that the number of true variables can diverge, but not too fast, that it scales at a lower order of n. So truly models with really unrealistically large sizes can have, we assume that they have prior probabilities 0, exactly 0. And under these assumptions, under this further assumption, we can take the sigma square out, and we can show that the posterior probability of the true model given the data converges to 1 as the sample size goes to infinity. So again, the posterior probability concentrates only on the true model. However, this is an asymptotic result, that as n goes to infinity, such things happen. But in finite sample, this is not guaranteed. In finite sample, we can have the posterior probability to be more diffuse, to not yet concentrate fully on the true model. So in practice, I always like to hedge my bets. In practice, we would like to kind of find all the models which, or rather as many models with high posterior probabilities as possible. That should be useful not only for inference, but also for predictions. So this is what we do is we developed a computational method called it's called SWEN. It's screening of variables with embedded screening. We couldn't come up with a better acronym. So, SWEN is my son's favorite character from that form of Disney film, Frozen. Yeah. I used to play a computer game, Dota, where Sven was one of my favorite heroes. So we chose this name, kind of. So let's suppose we have a model of model G, just to illustrate how we do the computations, how we find these models with high posterior probabilities. Let's suppose we have a model G right now. We can, let's say we start with this model G, right, which has two variables that we deem to be important. Let's say variable number 1 and 2 are deemed important, so they are in the model G. What we do is compute the posterior probability up to a normalizing constant of all the neighboring models of G. So all these neighboring models differ from G by 1 or 2 variables. So they can be the added sets of neighbors. Let's call G plus is the set of all neighbors of the model G, which has one more variable included. So 1, 2, 3, 1, 2, and 4, 1, 2, and 5, these are all in the added set G plus. We also consider the one deleted neighbor of G. So, we can delete 1, and that would give the model 2. We can delete 2, that would give the singleton model 1. And these were the deleted neighbors of G. And the swapped neighbors of G, called G naught. So where we replace one of the existing variables by a variable that's outside. So these are 1 and j for every j between 3 and p, and the models consisting of 2 and j for every j from 3 to p. Okay, so these are all the neighbors of G that differ from G by at most 2 variables. So G plus and G minus differ by 1 variable, G naught differ by 2 variables. Okay, so what we do, we consider this neighbor, and then we compute the unnormalized posterior mass of all G prime in the neighborhood of G. And this is a we won't be going into the details of the computation, but we have devised a smart Cholesky factor and quick like a Cholesky update that allows us to compute this G prime for all neighbors G prime of the model G. So we compute all these unnormalized posterior probabilities, and then we have this embedded screening step. So what we do is we keep only the best few models in the neighborhood of G. So we have all these posterior probabilities that are faithful to the Bayesian model, and we keep only the top few of them. And then we move to a new model among these top few with a probability proportional to their mass, to their posterior mass raised to the power 1 over T, where T is a temperature. And then we repeat for many iterations and multiple times, multiple temperature T that are less than log P plus log P. Now, why do we do all these things? So, and how do we, what happens? So, the computational complexity of computing all these neighbors can be proved to be to scale linearly in the number of variables P and the number of non-zero entries in Z. And remember, in our applications, we have these markers which are coded as 0 or 1. So this Z matrix is sparse. And so, mod Z naught is not n times P, which could be the maximum number of non-zero entries. But in general, the number of non-zero entries in Z for sparse Z matrix is smaller. So the computational complexity scales linearly as P plus norm of L0 norm of Z. And in some sense, this method is matrix free in the sense that we do not need to store any big matrix of, like P by P giant matrices. So the additional memory required, except storing Z, of course, is practically big O of the number of variables P. So it scales linearly with the number of variables. We screen using G prime and keep the best few models. That's the model-based screening to rapidly identify the models with high posterior probability. So there's the screening step embedded in the Bayesian variable selection. And we use this temperature control to avoid getting trapped in local maximum. So if T is high, it kind of flattens the posterior surface or posterior mass distribution so that this algorithm can jump from one mode to another, can escape from local modes. Because our goal is to find all, as many models with high posterior probabilities as possible. And we use this kind of temperature parameter because the theory, the posterior consistency theory says that log posterior probabilities of high probability models are separated by, big O of log P. So that's why we kind of have this flattening or temperature parameter. All right, so we can then proceed to in inference purposes, we can proceed in two ways. We can only consider the MAP model, the maximum a posteriori model, the model with the highest discovered posterior probability, or the weighted average model, which kind of, we have found all these models with high posterior probabilities, and we kind of choose only K of them, where, for example, which have like substantial posterior mass. And we just focus on those models, ignoring the rest, and compute the marginal inclusion probabilities based only on those high discovered posterior probability models. And then we can threshold this marginal posterior probabilities. Let's say the theory of Barbieri and Berger said that 0.5 is a good threshold, so we can consider the model to be the weighted average model to be the model containing variables J with approximate marginal inclusion probability exceeding 50%. We also have the posterior predictive distribution, averaged over all these gamma, but this is not tractable. So what we can do is, consider only these discovered high posterior probability models, and we can base our posterior predictive distribution based only on these high posterior probability models that we have discovered using this algorithm. Although this is not entirely tractable, but the mean and the variance can be expressed as expectation of some analytically available functions of gamma of the models given the data. And we can accordingly consider prediction interval, a Z prediction interval based on the posterior predictive mean and variance. How am I doing about with time? 10 minutes?
You've got 10 minutes.
Okay, thank you. Yeah. But we can also, do conditional simulation. So we can simulate from these posterior models, and then we can create a Monte Carlo prediction interval based on the quantiles of the posterior distributions. And we will compare those in terms of performance. Just a passing comment, is that our model is based on particular Gaussian structure, on the independent Gaussian structure, on the effect sizes and the model sizes. But it is possible to incorporate, the computational framework can be easily accommodated to some commonly used or new, priors on models. So, for example, the beta binomial priors on the model size, the determinantal point process prior on the model size, or Zellner's G prior on the, for example, on the effects on the coefficients. So, our computational framework can easily accommodate these priors on the models and coefficients. A real data example. So let's go to this Bayes data association study. We have close to 5,000 inbred lines and check varieties. So check varieties are those used. They're not always genotyped or sequenced. We don't have typically the marker information, but they're planted along with these inbred lines to have an understanding of the spatial variability. So among these 5,000 inbred lines, we have genetic data, the Z matrix, Z data for 951 varieties from Panzea project. We remove the duplicated SNP markers and markers with minor allele frequency less than 5%. And finally, we have more than 500,000 markers. That's the P. All right, so we first do a cross validation. How can we do how does prediction works? So we do a training and testing. We do a training set with 3,851 varieties and the testing of testing set of size 100 genotypes, and we relax the minor allele frequency to 1.5% in the training set. And we compared with Lasso Elastic Net and S5, which is the non-local prior by Shin et al. But the R packages failed to run on the entire data set with more than 500,000 markers. So we had to do a bit of a screening to just facilitate comparison with these other methods. Our method can run on the entire data with the entire set of markers, but others couldn't at that point of time. So we used the whole screening method and preserved the 3,850 markers, and then we trained using the model and then tested on the testing set and repeated this process 50 times. Now we chose different sets of tuning parameters just to see the effects, and on an average, what we see here is that the top five top five are the Bayesian methods. The bottom two are the frequentist methods. In general, what we see is the Bayesian methods tend to do in terms of prediction. So we computed the prediction errors on the testing set. They do a bit better on the prediction errors than the frequentist methods. But also, interesting thing is that they tend to have smaller model sizes than both Elastic Net and Lasso. And we also see that our method with high penalty, like more shrinkage, tend to do similar, on the prediction side, but they do with a smaller model size. In contrast, our method with low shrinkage does a slightly better on the prediction side, but has almost double model size than the high shrinkage methods. So that's with the prediction problem and model sizes. Our method can run successfully on the entire data. We have run Swen 50 times on the entire data set with a very high penalty on the model size. So we assume that W is 1 over P squared and lambda is like N over P squared. And we looked at the best models on these 50 runs. And as we generally think, like maybe the asymptotics hasn't kicked in, so the posterior mass is diffused still, and that was the case. So we have found many different models, but we have found among all these models the pair of markers that were never included jointly are highly correlated. So frequencies at which they are selected either in one model or in the other add up to 50 to the total number of runs. So for example, in this case, like the variable number 1, and let's say just 4, these are arbitrary numbers, just to enumerate, they haven't been included jointly ever, but they do share a 30% partial correlation between themselves after accounting for the remaining markers. So the model, like the algorithm Swen, can identify different set of markers which are possibly highly correlated among themselves. Finally, we studied the prediction intervals. Let's say we have, so we divided the entire data set into 2 parts with around 3,700 in the training and 200 in the testing. And, reducing the training set reduced the number of markers because some became duplicate. So it still has more than 500,000 markers. And we trained on this training set, and then we studied the performance of the prediction intervals on the testing set in 2 different ways. One using the Z prediction interval that is based on the posterior predictive mean and standard deviation. That's the ZPI. And the Monte Carlo based prediction intervals that does conditional simulations from the posterior models and uses the quantiles. So we use 10,000 samples from the posterior distributions to approximate the posterior predictive distribution. The coverage rates are presented here. So we find that 90%, the coverages are fine. So 90% confidence prediction intervals have like 91% coverages, 95% prediction intervals have 95% coverages. But when we look at the box plots of the widths of these prediction intervals, we see that the widths are more stable from the Z prediction intervals than the prediction intervals from the Monte Carlo methods. So by simulating from the posterior distribution.
5 minutes, Somak.
Yeah. All right. So that's all actually. So in conclusion, we have a fast and scalable Bayesian variable selection method for ultra-high dimensional problems. It's practically useful, and it also gives practically useful prediction interval for, let's say, if the agronomist come up with a new variety in his laboratory, breeds a new variety and wants to see how it may do. And so we can actually use a model to predict its phenotype without having to go through a season of planting and in the field and growing in the crop. The package is available on CRAN as a part. So the algorithm is available on CRAN as a part of the Bravo package. And we are currently working on extending these methods to GLM to accommodate RNA-seq data that are continuous covariants and can have nonlinear effects on the phenotypic response. And in many of these cases, we have functional linear models. A PhD student is working on this on actually the second project. And in these methods, what we assumed is, we did a we still did a two-stage method where we first accounted for the field design and spatial effects using statistical model and computed the blues. The blues were subsequently used in the Bayesian framework. And sometimes actually we are just provided with those because the scientists just compute the blues and then they provide that with the blues. But in many cases, we do actually have the raw data with the design information and with the design information. So we can actually extend these models to be like a one giant Bayesian hierarchical model that accommodates spatial and design effects and other type of random effects. And we are also working on extending these models to incorporate interactions among the genotypes. So yes, interactions among the genotypes, not only that, interactions among the markers. So that's all. Thank you for your patience. And these are the references. Thank you.
Excellent. Thank you so much. I never remember how to do the applause emoji, but there you go. I can see plenty of them. That was great. Thank you. I think I'll stop recording there just because that's an hour. Yes.